Welcome, everybody. So today we've got Bhavin Khatri from Imperial and the Crick, who's going to tell us about adaptation, entropy, and evolution.
Cool.
It's yours.
Thank you very much. So I guess this is not an evolutionary biology audience. And so I guess I should define what evolution is to begin with. And the first thing you want to ask is, what is life? And since you are a non-equilibrium statistical physics audience, this is probably my best definition of what life probably is from a physics perspective. And that is, it's a driven, takes in energy, non-equilibrium system with some sort of emergent properties of memory and prediction. But what we have now is Darwinian evolution, which we can characterize by reproducing organisms as individuals that have DNA or information about how to survive in a given environment. And so selection, natural selection, is those organisms that have the best information about how to survive or reproduce in a given environment, survival of the fittest. Mutation is random changes in that information. Mutation plus selection is adaptation to the best information. And then this final thing, which most people don't really consider, is random genetic drift. Stochasticity due to the finite size of population. So sometimes populations adapt to suboptimal information. Why has that not come up? Oh, here we go. So variation happens by mutation in our genotypes. Yep. So we have in our cells, we have DNA, which is codes for a particular phenotypic expression. And if there's a mutation at a given point, that might change the phenotype in some way. So I'm going to have a relatively pedagogical introduction to evolution and natural selection. Just I don't know what the audience is here. And so natural selection says there may be a selective advantage to one variant. So it may leave more offspring on average. And so if you imagine a population of individuals that have these different alleles, red and green, they reproduce, they leave the offspring. We have finite resources in our environment, and therefore we assume there's a fixed population size. That's the simplest model. And so when we resample the population for the next generation, this is essentially the basis of something called the Wright-Fisher model. We have, on average, more greens left because they left more offspring. And this process goes on and on over many generations till you're only left with the green allele, okay? Because it has selective advantage. I think that's going to be relatively obvious to most people. But this green allele has to arise in a single individual, okay? And then it has to somehow survive to go on. But it could be unlucky. It could be at some point run over by a Maserati in this case and be lost. And so genetic drift, I'm going to come on to what this phrasing drift, because it's a mathematical physics audience, but is randomness in the evolutionary outcomes, okay? So sometimes good mutations are lost. And so what they need to do, these mutations have to establish in this population before they can actually manifest their selective advantage. So this is a bunch of replicate simulations starting at 1 over n in the population. And I'm not sure you can see the colors so well, but the cyan green are those that are lost immediately, or before they actually even get up to large frequency. And the ones which are in red are the ones that actually survive the stochastic process at the beginning and go on to be selected and then dominate the population, go to fixation. So this is allele. So this is allele frequency of that green variant, say, where it has a selective advantage. And it starts off at 1 over n in the population. There's n individuals. Yeah. And so it has to, yeah. And so it turns out that it has to get past this frequency of 1 over 2ns, this critical frequency, in order for it to actually establish in the population. So there's a stochastic phase, and then there's a deterministic phase, roughly deterministic phase. The selective advantage. And big N is the population size. And so a few years ago, Desai and Fisher analyzed the branching process of this thing. When you're at very small frequencies, there's a branching process you can write down, and you can show that actually the allele frequency does this sort of thing, i.e. at in order for these, so if you only look at the, this calculates the mean frequency of those alleles that are destined to establish. Yep. So if you only look at the red curves, and we calculate the branching process for that red curve, these red curves that are destined to, conditioned on establishing, then we have the mean looks like this, which is this black curve. And so those guys that are destined to establish have to be lucky enough that they grow faster than selection in order to actually be established in population. And then asymptotically, for large times, we then get this behavior here, which is exponential. Right, so if we go away from the branching process, we're not at very, very small frequencies, and we can make essentially a continuum approximation. We have what's called the diffusion approximation, which is the equation motion for genetic drift, okay? And like I said, I'm going to go into why it's called drift and why maybe it's not the best name, of course. So here we have a Langevin equation, which has this white noise term eta. And there's an effective diffusion constant, which goes as x1 minus x, right, divided by n, the square root of that. And that looks something like this. So basically, diffusion becomes more and more difficult as you get towards the edges of 0 or 1, when you're near fixation or when you're when you're near extinction. So this is difficult to deal with. X-dependent diffusion constant, although Kimura found the series solution many years ago. So the question is that evolution biologists have used this term drift, and we know in a mathematical sense this is slightly bizarre. But so go on. Yep. No, sure. So you can derive it phenomenologically. You can derive it from some underlying master equation and do some sort of Van Kampen type thing. But essentially, so the way it's derived is as a Fokker-Planck equation. Yep. With a mean and essentially you calculate what the mean change in frequency is, and you know what the variance is, and you can calculate the variance from this Wright-Fisher type process, and it gives you this thing. So when there's no selection or mutation, this is what you have. You can't, no, yeah, sure. You can't fluctuate either. Yeah, exactly. And so you can argue with this from looking at the binomial distribution, basically. Yep, for your transition probabilities. I am, well, sort of. I already have in a way. I wasn't going to go into the details, but so yeah, okay. So maybe I should say the Wright-Fisher, like I said, so the Wright-Fisher model is a model where you say you have a fixed population size of n individuals, and then you resample your next generation. This is a pseudo generation in some sense. You resample the whole of your population by essentially doing binomial sampling. Yep. Of that. And then that generates the next generation. So if you have a frequency P, in the next generation, the mean, the expected frequency is going to be P again. But it's a random, you got binomial sampling, so you know the variance goes as 1 over n. Yep. And so large populations don't have much drift. or it takes a very long time for it to act. But small populations have significant drift. And this is essentially what this is, a Langevin representation of that, or a stochastic differential equation. Yeah. Large N continuous time. Yeah. Right. So the individual trajectories themselves have no bias, right? So the mean, like I just said to you, yeah, the expected mean is whatever your initial frequency was in the next generation. They have no bias, so clearly it can't be drift in a mathematical sense. But the mean squared displacement has this property here, which goes as x1 minus x, and has a position dependence. And so if we look at at any given point, the size of the jump changes, but it's equally probable to go up in frequency or down in frequency, and so on and so forth. And this is true for any Ito process. So the Wright-Fisher and even the Moran process are Ito processes. But you can have a flux, right? So although the individual trajectories have no bias, there's a net probability flux towards the boundaries because the mean square displacement depends on x. So we imagine taking some arbitrary plane, x. We ask what diffusers are coming from the left, from a lower diffusion constant, say, if this is x less than a half, versus all those diffusers coming from the right. There's a larger volume that will pass this point per unit time, and therefore there's a net flux, in this case to the left, I guess. Yeah, and that gives rise to fixation or loss. Yep, and there's a flux of an ensemble of random walkers. You have a flux going towards the boundary. So it turns out that Ronald Fisher found a way of simplifying this equation by coordinate transformation, which is just theta cos to the minus 1 minus 2x. And what that does is it gives you a new coordinate, a new Langevin equation or Fokker-Planck equation, which has coordinate independent diffusion. Okay, which is nice because then we can actually then, use some more tools that we know about stochastic processes. But what's happened is that we didn't have a drift term here, but now we have a drift term which represents this flux in the normal space. Okay. So we have coordinate independent diffusion, 1 over n. And so this genetic drift, right, gives mathematical drift in this angular different space, angular space. So a better name would be, which I propose in this paper, genetic flux. And this is an example, population genetics is an example of a flux without drift. And they talk about various different types of stochastic processes in that paper. And in the same paper, I found a way of finding Green's function of this. I'm not going to talk about that now, and including selection and mutation. So then you can actually apply that to real data, trying to determine selection versus drift and things like that. Right, so let's now try to step back a bit on longer times. So a very important quantity, of course, is the mutation rate multiplied by the population size, which tells you the number of new mutations entering a population per generation. If that's large, then we have these sort of clonal interference type dynamics. There's multiple variants competing at the same time. But if the mutation rate multiplied by population size is much less than 1, we have the sequential evolutionary type process. So the process of drift, fixation or loss happens much more quickly than it takes for the next mutation to be generated in the population. Well, Muller's ratchet applies to this, yes. This scenario, yes. Yeah. And so then using this diffusion approximation, you can calculate a probability of fixation given the fitness of a mutant in that, in this case, starting with the frequency of 1 over n. And that looks something like this. So this is for positive positive fitness advantage. So now I've used delta F instead of S, but they mean the same thing. Sorry about that. And so, you can have positive changes, you can have negative changes in the fitness of the mutant. And for those changes which are much less than 1 over n or minus 1 over n, they're effectively neutral. Yeah. So this is the probability fixation on a log scale. And if n is the population size, then you expect a neutral variant to have a probability fixation of 1 over n, because they're all identical and they should all have the same probability of fixing.
So delta F is what we call S before.
Is what I called delta F is what I called S before.
So it can be negative.
It can be negative. And it says that, sorry?
What is negative?
It means it's deleterious. It's not.
So you basically have
You mean negative delta S? Yeah.
So this thing is it? Can you say that the typical value of delta F is negative?
I'm actually going to come on to that later. It depends on the system, right? Depends on the, yeah. Not quite. Let me ask, I'll allow you to ask that question again if it hasn't been clarified later. And so on a broader scale, if we imagine there's some sort of fitness landscape, okay, and has some singular peak, then what this picture tells us, genetic drift, at least at the moment, it seems it's just telling us that we only have fluctuations of order 1 over n around the peak. We still adapt to this peak. There may be fluctuations on the way, but we essentially adapt to some peak, and there's as we decrease population size, the size of that variation will change, of course. But importantly, the change in the phenotype, so this is the phenotypic axis. The change in the phenotype only goes as 1 over square root of n. It's just a fluctuation around some Gaussian-like peak or quadratic peak. So the scale of changes in phenotype are only of order 1 over root n. And so we can do this sort of thing. We can say, okay, we think there's a certain trait which is optimal, and therefore I'm going to have a fitness landscape which tells me that that is optimal. We could even have a rugged spin glass type landscape, and say we think some process is rough, the evolutionary landscape is rough. But if you do that, there's no possibility of an emergent phenomenon. You essentially get what you put into the thing, right? So you want to get something, you want to be surprised by the answer, you want to have the answer to emerge rather than you putting the answer in. And so this speaks to my title, which is, in some sense, this is like the adaptionist viewpoint, that if you say, I think this phenotype or this thing is important for some reason, you've already given yourself the answer. There's no possibility for anything emergent to arise. Uh-huh. Yep. Uh-huh. Yep. Yep. Okay. So I'm talking mainly from evolutionary biology perspective, and this is typically the sort of reasoning and views. But in that case, I mean, I'm going to talk about a case of developmental biology and some sort of gene regulatory network. But So in this case, are they looking at evolution or are they looking at developmental process? Yeah. Right, sure. Okay. So the question is, yeah, sure. So, but the question broadly would be, is there some sort of, if you're doing evolutionary dynamics, are you imposing a fitness landscape in some way that will give you the answer to begin, you expect? And so that's what I'm talking about here. Right, so let's go back to this picture of genotypes and phenotypes. So the key thing is that mutation and variation happen at the genotypic level. This is where mutation arises, yep. And somehow it's translated into a phenotype through some complex mapping, and selection arises at this level, okay? So that's an important distinction to make. And typically these genotype-phenotype maps are expected to be very redundant. So this is where statistical mechanics could play a role. And I'm going to talk about that now. And so here we've just got some arbitrary mapping to phenotypic space. There's like a cartoon picture, and then that maps to some fitness. And you see there's many, many genotypes that give you a particular phenotype. And one,
when most of the stuff we've been studying in this program has been about looking at things and but we're not talking about evolution, we're talking about something.
Sure. Yeah. No, sure.
Much longer.
Very long time scale.
So,
yeah.
For bacteria, it's not so much longer.
Well,
it's
yeah, it's true.
It's quite long with the life cycle of the bacteria.
I mean, I think if you're looking at active processes within the cell, that's determining the phenotype of that cell. So there'll be mutations in the things that code for actin and myosin and other, proteins that, yeah.
So I guess my question is just getting the sense of the time scale. So typically, I know that for the individual, we're talking usually the time scale of many generations. But can we think of it even at, you're saying that you could think of it even at a cellular scale where it might be a shorter time scale? That you can, are you saying that?
Well, I was saying there that the way mutations that arise in a particular way, it would change the phenotype of the way actin and myosin networks work. That, depending on the fitness advantage or disadvantage, the dynamics of how that plays out will take many generations. Yeah. And typically you don't expect fitness differences are normally of order a few percent. Except for some like extreme like genetic engineering cases possibly. But yeah. So clearly the mutations must happen at an individual cellular level, yeah, within the genotype. And it's expressed in some way in terms of what changes the dynamics, but how that plays out can take a long time. Yep.
I guess, what's the rule of sexual? Do we happen on the level? So.
I mean, I haven't said it actually. I'm essentially assuming asexual random reproduction. Yep. All of this. Yep. Sure. Well, no, well, yes and no, but still wrong. Oh, there you go. That's true. Yep.
But that's the process of recombination. That's a particular.
Well, I mean, so that's at a somatic level. Yeah. So that's, yeah. As in within the immune cells and yeah. Yep. Sure. Yeah. So I'm mainly talking about germline evolution, but yeah, it's the same principle though. But it's not a distinction you really need to make for the broad sort of things I'm talking about. Okay, so if you look at, so it's talking about redundancy of the genotype-phenotype map. And so if you look at the database of all proteins that have been crystallized, on the right, we have the, for example, here. So these are a different number of distinct protein folds that have been found, which is in green, sorry, here. And these are the total number of sequences in blue. And we see it far outnumbers the number of actual number of folds. So there's a redundancy between these two mappings. So you want given this redundancy, it naturally the sort of question arises: what happens? What's the role of this redundancy in evolutionary dynamics? So imagine this toy scenario where each point here is a genotype, and we've colored it by its phenotype. Okay, it's different colors, and they have different fitnesses. So, for example, the blue one has fitness 2, it leaves 2 offspring, and the pink one has much higher fitness of 7. But there are fewer sequences, fewer genotypes that code for it. Okay, so let's call omega the number of genotypes. In this case, it's 52. In this case, it's 4. Will the pink phenotype always win? Is the broad question we want to ask. Now, I'm going to now try to ask that question with a concrete model based on the biophysics of transcription factor DNA binding. And so we have a transcription factor that binds a bit of DNA to turn on transcription of some other gene. So it turns out amino acids typically have a single preferred nucleotide they like to bind to. Non-preferred ones cause a large destabilization in the binding affinity. And on top of that, once a binding site has of order a few mismatches, the non-preferred amino acids, then the transcription factor no longer binds specifically, but it changes conformation and has an electrostatic mode of binding. So the simple model I'm going to talk about is one where we say that the genotype of the transcription factor is just only expressed by the nucleotide it likes to bind to. So amino acids are just only have 4 flavors now, okay? And then we have our DNA, which already has just 4 types. The binding energy is just going to be equal to some energy scale multiplied by the number of mismatches. So epsilon is typically of order 3 kBt. But then we can enumerate here, we can explicitly enumerate the number of sequences that give me a particular, this case, phenotype, which is the binding affinity or the number of mismatches. And it just goes, it's just a binomial distribution, okay? So we can do a Monte Carlo simulation of this sort of process. We have a genotype, we can calculate the number of mismatches. We assume some fitness function. So I am now assuming some fitness function and making an assumption that it's good for it to have high stability, right? Or low binding energy. And I've chosen this quadratic form. And I've got this truncation here beyond a certain number of mismatches. It becomes completely deleterious because it's a non-specific mode of binding. I can calculate the change in fitness. I can calculate a fitness, and therefore I can calculate a change in fitness, and that tells me the probability of fixation of a mutant. And then I can keep going around this process and do an evolutionary simulation. And if we have a completely neutral simulation, what we find is that the binding energies are peaked towards very unstable binding energies, as you might expect, right? So there's a large peak out of very, poor stabilities. And this is essentially what you find if you look at, there's a lot that goes into this curve, so take a slight pinch of salt. But if you take the preferred binding pattern of a given transcription factor and then just scan it across the whole genome of, in this case, I think it was yeast, you get essentially this curve here. The black curve here is exactly this one, but on a log scale. So it's this binomial distribution. But as you see, there's a deviation at small binding energies, and that's because that's exactly where selection's acting, okay? And then you get a deviation from the neutral distribution. So the key thing is our neutral distribution is highly biased to begin with. So if you start thinking about evolution of this phenotype, you need to consider the underlying neutral distribution because of this genotype-phenotype map.
So
Oh yeah, yeah.
so there's going to be some. When L is so typically L is relatively small, short. So
L is of order like 10 to 15, I think,
when L is
roughly.
large,
Yeah.
yes.
But even for 10, it's pretty Gaussian. Yeah.
so
I mean, it's essentially, yeah. And so, yeah, exactly. Here.
Same as in the mixed plot, right? But then you had
no. I think the inset.
the inset, there was also a little.
There's a histogram. I presumably that's just staying.
Oh, okay. But you have to, so this is I can't remember now. It's not my paper. It's not my paper. It's not my paper. No. I think it's basically so this is them from Moscone and Michael Lessig, and they basically try to determine some fitness landscape of transcription factor DNA binding from looking at the neutral distribution versus the actual distribution, and then it gives you that. I think that's what that is.
That's something inferred from that.
Yeah. So now if you turn on selection, and I've got my strength for selection is kappa F, which is the curvature of my fitness landscape. We see that at small population sizes, we just have this neutral. All right, let's start at a large population size. The large population sizes, when this thing, the normalized fitness by population size is much larger than 1, we get what you'd expect, right? You adapt to the best binder, yeah, the most stable binder. But as you decrease the population size, we see the mean shifts, and we get to this essentially neutral distribution, which is the tail of the one I showed you earlier, because I have this sort of truncation selection. So how can we understand this in a quantitative way? So in equilibrium, if we assume some sort of microscopic reversibility at the genotypic level, detail balance will be obeyed. So if you imagine there's no, the mutations go either way and they're equally likely between two different genotypes, they may have a difference in fitness, then we can use a detail balance argument to say that the flux between those two must be the same. And this phi is the probability of fixation, as before. And then the ratios of those two turns out to give you a Boltzmann factor. So the equilibrium distribution of genotypes is a Boltzmann distribution. Making this assumption, so it's important to realize the assumption I'm making here is that we have some sort of equilibrium type evolutionary process, right? So the fitness landscape or the fitness is not changing over time, okay? And this is actually shown by a couple of people in the past, Iwasa and Soder and Hirsch previously. So we see that here 1 over the population size looks like KBT, and like I said before, differences in fitness of less than 1 over 2n, or 2n in this case, are neutral.
So what is assuming here is that so that you consider the landscape to be station on the scale of where is I don't why would you say that?
So it's equilibrium. So I'm just, if you assume the fitness landscape doesn't change over time. Oh yeah, so I should say that for something like transcription factor DNA binding, that might be a reasonable assumption. It may not be. I'm going to talk about protein stability in a second. So then over a very long time scale, you expect it to be that fitness. So there's a term called stabilizing selection. Yeah, that the selective pressures are not changing over time. If there's adaptive changes and there's certain traits to responding to more local changes in the environment, then this sort of thing won't be true. Yep. Okay. So I'm essentially saying that the environment is fixed and the pressure changing for this transcription factor to turn on something is not very large, yeah.
So I guess component 100%. But what you're saying is that, again, I'm trying to understand.
Yep.
So you have this idea, we have this idea, I think of it maybe there's some selection, there's some kind of background selection pressure or something.
Yep.
Environment is changing. And then when that's happening, this kind of equilibrium works. Then there will be some change. Let's say there's a famine or something or drought.
Yep.
Then there's going to be a new.
Yep.
And then I'll have a new landscape. Then I'll have to.
Yeah. So in that, if it affects this particular trait we're looking at, then this argument would not work, right? There'll be some fluxes in the landscape, and so there'll be non-equilibrium. But the way I think about it is that there's, organisms under all sorts of different selective pressures. Some traits will be under very short time pressures. Other traits are so important that effectively, even though the environment's changing, there's some sort of buffering, which means that those traits are effectively stationary. I mean, the effective requirement is changes very, very slowly. In practice or in the model? In the model, it's just, you have where did it go? Here we go. So this is just a string, which you have in your computer, and you say you could start it off randomly if you wanted to. You just count the number of mismatches. That's it. They always assume the equal length. Yeah, exactly. It's just two quaternary strings in this case. Yeah. Right. Okay. So we have the fitness, the distribution of genotypes is Boltzmann distributed. And so what happens to the fitness of phenotypes? So this is our Boltzmann distribution. We want to sum over, we have some arbitrary genotype-phenotype map. Let's just represent it in this way by this function. And so we want to sum over all our genotypic states and then have a delta function to pick out when we have a particular stability equaling our actual function here. And that then is going to, relatively simply gives us some degeneracy factor. So for a given phenotype, we have e to the power 2n f of that phenotype, and then multiply by the degeneracy factor. So how many genotypes give me that particular phenotype? And so you can express that degeneracy as into the exponential. And what we have is we can express this as a free fitness. So we have the fitness plus an entropy term divided by the population size. An entropy term is just a log of the number of genotypic states that give me that phenotype. So here in this, again, I stress this in this equilibrium assumption, you have this analogy to the free fitness, where the potential energy goes as minus F, the free energy goes as minus the free fitness, and KBT is 1/2n in this case. And so now if you calculate exactly what that is, it's these circles, and they match the values of the simulation to the squares perfectly. So that works well. So at large n, fitness dominates. At small n, phenotypic entropy dominates, and you have something in between. The populations generally will adapt at any finite population to a non-optimal phenotype. Okay. And importantly, the shift in the mean phenotype away from the optimum is now of order 1/n. So it's a much stronger effect than just looking at fluctuations around some fitness peak. Let's now look at another example of the stability of proteins. So if we look at stability of proteins, look at a database of stability of proteins, and we find that it's peaked at roughly -5 kilocalories per mole. And so, but the free energy of a single hydrogen bond is only of order a few kilocalories per mole. Okay, so there's been this outstanding question of why aren't proteins, why are they only marginally stable? Okay, they could be far more stable. And actually, I've forgotten to put the references here. People have actually engineered proteins to become more stable. They can be done. So why aren't they? Maybe we've forgotten about some trait which is providing some sort of trade-off. But maybe proteins need to be floppy. Okay. And so in our fitness landscape, we need to have another axis, or it could be multiple axes. And then that's why we're not getting to our maximum stability, yeah, because there's an intermediate peak. So this is strongly argued against by, and this is many, many years ago, by Stephen Jay Gould and Robert Lewontin, both evolutionary biologists. It was a very flamboyantly named paper called The Spandrels of San Marco and the Panglossian Paradigm, a critique of the adaptationist program. So let me quickly read this out. How much time have I got? Okay, very quickly read this out. The adaptationist program has been dominated thought in the England and the United States for the past 40 years. It's based on the faith in power of natural selection as an optimizing agent. It proceeds by breaking an organism into unitary traits and proposing an adaptive story for each considered separately. Trade-offs amongst competing selective demands exert only a brake upon perfection. Non-optimality is therefore rendered a result of an adaptation as well. And then they go on to criticize using various examples. And I'll just talk about one, the ones they use, which is the Spandrels of St. Marco. So we have these structures in churches, which are very nice and beautiful. Yep. And so people think they must have been designed from the get-go. They must have been architecturally designed because they're such nice, amazing things. And we can put interesting iconography in them. But if you just consider, if you want to put a dome on 4 arches on a square, you will have a spandrel. Yep. You have this triangular type shaped structure. And then afterwards, because it's there, then you'll use it to put things in and to put iconography, etc. So there was no, in the analogy, there's no selective pressure for spandrels, but then they were co-adapted later. Another example is Archaeopteryx. There's still lots of debate whether it could actually fly or not, but it has feathers. So the adaptationist thinking would be, it has feathers, therefore it must have flown. Yep. But the alternative argument is that feathers were used for thermal regulation. They first evolved for thermal regulation, and then it gave some advantage in, proto-birds falling, and then it eventually led to flight. So again, so you need to look for more structural sorts of reasons. And in the case of protein stability, we go back to this plot here. We have this redundancy I mentioned before. And if you look at, do simulations of proteins fixed to a particular fold, and you look at random sequences, our null distribution then becomes, again, like the transcription factor DNA binding case, highly peaked towards poor stabilities. Sorry? So, okay, we've gone through it quickly. So if you take this particular protein fold, yep, you fix the structure, and then you say, now let me look at random sequences. Of the protein. So we have this structure. So there'll be a fold in nature. Yeah. And you ask what's stability relative to some unfolded state. And it gives you this, again, this null distribution, which is highly peaked towards poor stabilities. So again, we have the same picture that natural selection will be taking one direction towards more optimal binding, but genetic drift will take in the opposite direction. And that balance is, like I said, will be mediated by population size. And so this predicts that as you decrease the population size, proteins should become less stable. The population of individuals, yeah. Well, it depends on what question you're asking. Yeah. So it could be a population of cells if it's bacteria. Yeah. If you're asking what's the evolutionary process of a collection of bacteria or a particular species of bacteria, what's its population size, then that's what would go into this. But if it's higher, eukaryotes, us, then it's the population size of humans or population size of birds, or yeah, to go into this. All very slow. And so if you look at, again, you can look at a database of proteins and the effect on stability of random mutations, and you find that it's peaked towards less stable mutations. You generate more unstable mutations on average, which is exactly what this picture. This is so slow. Yeah, so this is telling you that you're going to generate more if you generate the distribution of stabilities, changes in stabilities of proteins, if you generate them randomly, will be biased towards being more unstable. Yep, and this is what you find. Right, okay. And now if you do simulations, so two different groups have done similar simulations of what happens to stability of proteins as a function of population size in this population genetic process, and you do find that the stability does go down. Stability goes down as you decrease population size, or the free energy decreases the increase in population size. Both these models. And I did find one paper which tried to compare the stability of proteins in prokaryotes versus eukaryotes. They found that prokaryotes are generally more stable, and they make the adaptationist argument here, saying that results show that kinetic stability is prevalent in prokaryotes, especially thermophils, but uncommon in eukaryotic organisms, thereby suggesting that this property might be crucial for the adaptation and survival of less complex prokaryotic organisms. But I would suggest it's just a fact of a general mapping from genotype to phenotype. There may be an adaptive argument, but you have to prove beyond the neutral distribution that there's a deviation in order for you to make that claim. Okay, I think the next part, yeah. Okay, I'll just go very, very quickly through this. And so what are some of the consequences of this genotype-phenotype map for evolutionary processes? So one thing you can do is look at a model of speciation. You assume you have some sort of common ancestor, so there's some sort of split. So then you have two divergent lineages. Then you can have evolution of both this transcription factor and DNA binding site. They co-evolve with each other on each of these lineages. And then you can ask after some period of time, what's the stability of hybrids? Yep. I can create a hybrid and ask, does the transcription factor from one lineage still bind to the binding site of the other lineage? If it still binds well, then it's basically essentially one organism. They haven't split as a species. But if it doesn't bind well, then effectively what you have is what's called a Dobzhansky-Muller hybrid incompatibility, and that gives you full binding, and it's assumed that will then lead to a deleterious change in the organism. I'm only waiting for the finger update. Here we go. Okay, so if we calculate the probability of incompatibility as a function of divergence time, we see that smaller populations give you more incompatibilities more quickly, and larger populations takes longer for them to arise. And you can understand it within this model because so essentially you can understand it within this model because the common ancestor is already going to be somewhat maladapted, and therefore it leaves less substitutions in each of the lineages before you get incompatibility. Okay, here we go. because so yeah, so it's these things arise more quickly, and it's power law distributed because you're very close to the boundary, and you have these substitutions that are Poisson distributed. It's so very slow. Okay, anyway, so for large population sizes, you get, because the common ancestor is further away from the boundary, it takes more substitutions and you get this sort of behavior. I'm not sure I have time to go through all of this. Okay, so how can we rationalize this sort of thing, this sort of behavior in terms of stochastic processes we know? So can we write down the stochastic dynamics for phenotypes, accounting for this sort of entropic pressure from sequences, the mapping of sequence to phenotype, genotype to phenotype? Remember, selection acts on phenotypes, variation arises at the genotypic level. So is it normally this slow?
I think Zoom has a delay, but it's because of Zoom. Whenever the call is.
Oh, here we go. Right, so we can imagine writing down a master equation for how the phenotype changes over time. And we have some function that tells us what the rate of change from two different phenotypes is in going different directions. We can make the assumption that the changes are local. Did it come up yet? No. And then we essentially have delta functions for local changes. Wow. Maybe it's my laptop. Here we go. Let me just, yeah. Sorry? Yeah, but I've got, yeah. Anyway, so we can make the assumption, and so we can go through the whole process and write down some effective stochastic dynamics. You can do that, but instead I'm just going to show you a heuristic argument. So right, shall I leave it? Do you think? How do I leave the meeting? Oh, you can't see the animation though. I think let's just power through. It should be okay. Oh no, we just. Right, so you can imagine some sort of fitness landscape as a function of phenotype. There'll be some entropic landscape, and then we can calculate some effective free fitness landscape by combining the two. And then we expect fluxes to be proportional to the derivative of this free fitness landscape with respect to our phenotype. And we have some effective friction or mobility mediating that. The question is, what is this effective friction or mobility? And you can use the Einstein type argument to say that the flux due to drift must balance the diffusional flux. And we know what the distribution is in equilibrium. It's just a Boltzmann distribution. And therefore you get that this effective mobility or inverse of friction is just n times mu. And then you can write down a Smolotowski equation which describes phenotypic evolution accounting for this sequence entropy or this phenotypic entropy effect from the free fitness. And so the previous approach gives essentially the same result, gives you the same result. And then we can write down a Largevin equation for this. this. And just white noise. And for multiple traits, you can write down multiple Langevin equations, a couple of Langevin equations, and then we have this noise structure. And so in this case, Gaussian noise represents random mutations. Okay, I can talk about that more later in this first order Markov. And so essentially what I want to do, I'm going through this relatively quickly, is to try to apply this to this transcription factor DNA binding problem. If I think now, let's just replace these with binary sequences. And then I'm going to replace these sequences with just scalar variables. Okay. And so my binding affinity is just the modulus of the difference between these two scalar variables. But the key idea is that selection acts on phenotypes, and therefore what we need is the correct distribution of phenotypes, and that's accounted for by the high dimensional properties of this Hamming space, which is through this entropy function S of xi. And in this case, if it's binary, we just have the binomial coefficient, which looks roughly Gaussian. The peak at L/2 in this case, and then our entropy function is just a quadratic. This is what the overall landscape looks like, but essentially, if you assume we're confined to just one of these peaks, it's effectively just quadratic. I'm going through it relatively quickly. So then essentially what we can do, our co-evolution of sequences is just Brownian motion of two bees connected by a harmonic string, but that string has a elastic constant, which is a function of population size. The solutions for x1 and x2 are just going to be Gaussian. And so we can look at this process and replace them by effectively springs. Now these things co-evolve independently from these. So we have now 4 variables, which are the effective sequences on each lineage, and then the primes are the sequences on the second lineage. And then the difference between these two is its effective binding affinity. And we can look at crosses, and then what we have is a stochastic process for these 4 things. And what we're interested in is the binding affinity of the hybrids, which is what's the difference between x1 and x2 prime and x1 prime and x2, which gives us these 2 different hybrid binding energies, and you can go through this stochastic process. So if it's incompatibility, you can calculate that if it's beyond a certain distance. So some of these things are Gaussian also. And so from that, you can calculate what the mean square displacement of the hybrid binding energies is as a function of time. So I've gone through this very quickly. And you get this 2 mu t type thing, which is quite nice. And then, which is due to inter plus intralineage divergence. And then once the intralineage relaxes, you only have the divergence of those 2 sequences. So you can calculate the probability of these incompatibilities in this model, and it gives you this sort of behavior we found at large population sizes, which has this root t e to the 1 minus 1 over t type behavior. So because the hybrids have to effectively diffuse a large distance, we don't get power law, we get this sort of behavior here. So the intuitive way to think about this is that on each of the lineages, we have co-evolution of these 2 sequences, which constrains the diffusion process to this like tube. And that's true on both lineages, but the hybrids don't have to obey that, and the hybrids just diffuse spherically, symmetrically, and it's just a question of finding how long it takes to reach the boundaries. And if you start with a smaller population size, you're nearer the boundary, and therefore it takes less time for incompatibility to arise, and you have these things, this behavior. So incompatibilities arise, speciation arises more quickly for smaller population sizes. Okay, and then you can apply this to a more complicated model, and essentially you get the same sort of behavior. I'll get through this very quickly. So this is just, yeah. So if you have a model of patterning an embryo, we want to have a sharp profile, and we express our binding energies and our proteins and their sequences by binary sequences, then we get a very complex fitness landscape. It's quite surprising given that you have a few transcription factors, and then we have a similar thing where there's low entropy solutions, with high entropy solutions. So there's many. So each one of these dots is a point in effectively a genotype or a position visited by this in the simulation. As we increase the population size, we get restricted to the higher peaks. But low population sizes, we have all these different sequences down here, which you need to contend with. There's high entropy, there's low entropy. Oh, yeah, and so higher morphogen gradients will give you a higher fitness because you can do sharper patterning. And then we can plot this free fitness as a function of morphogen steepness. And we see at low population size, we only have a single peak just dominate because of this high entropy. And a large population size, we have this bistability, where you adapt to the fitness peak, the one you would expect to be the best. Yep. And then you can do the same thing, the same model, you get the same prediction that speciation happens at more quickly for low population sizes, even in a small complex model. Right. Summarize and possibly point towards a universal principle. So we got this genotype-phenotype maps, and one way to characterize that for this stationary evolutionary process is through a free fitness function, which is analogous to the free energy in canonical ensemble. We have a balance between phenotypic entropy and fitness, where the balance is determined by the population size or the inverse temperature. So importantly, adaptation is not all. So you need to account for this before you start claiming there's adaptation going on. So my guess is for many types of phenotypes, which are these stability-based phenotypes, this will be a general principle. Because the most fit sequences, the ones that have the most stability, will be the most specified, okay? And they will have the fewest sequences. And so we will always have this competition between fitness and entropy. I'm not saying it's going to be true for all phenotypes, but I think, yeah, at least in this one, I'm reasonably confident this should be a general principle. So the picture we should have really is that the evolutionary dynamics is on these neutral planes of fitness, determined by this balance between entropy and fitness. And then we have these neutral networks, which people have previously talked about. And in this case, it suggests that the size of the neutral networks depend on the population size. And going forward, so the broad predictions that the size of the neutral network should scale as with population size. So how does this impact the rate of evolutionary innovation? And so Andreas Wagner is one of the people who've strongly put forward this idea that actually neutral networks are really, really important for evolution because they give you robustness, of course, but that robustness allows evolvability because then it allows you to diffuse to different parts of the network, which can then lead to adaptations. And as we're talking in a non-equilibrium physics workshop, I think it'd be interesting to explore these sorts of analogies, these evolutionary analogies, on models where we don't make this assumption that things are fixed, but there's adaptation going on. And the most obvious ones where there's some sort of breaking of time reversal symmetry. For example, red-green dynamics by antigen-antibody evolution, where there would be no fitness landscape which is fixed and it's continually evolving and changing. Cool, that's all I have to say. Thank you.
Thanks, Bhavin, for a really thought-provoking talk. Any questions, comments? Okay.
I was wondering, particularly in the bit where you were looking at changes in the DNA and that then leading on to changes in the folding of proteins and stuff, and those being passed down. Do you have any thoughts on that? How much that would be affected by the selection going involved into reproduction, like through like the spermagenesis, folliculogenesis, like producing the germ cells, and then the actual, you know, reproductive process, because that surely would then also, if these mutations were bad for fitness of cells, they would be unlikely to be passed on to the next generations because of those processes as well. So you've got like double selection or something.
I'm not quite sure I'm completely following. So, I mean, this is somewhat simple model. It says if there's any change, mutation in the stability of the, say, the protein, there's a chance it will be fixed because of this process, but the most likely thing, it will just be lost because it can't propagate in the population. It can't be passed on. So
I just think there like less chance of you getting, because it looks like a lot of, it's like a high probability that you get a negative impact.
Yeah.
Oh, and would that be reduced by the fact that if it was negatively impacted, sperm, say, wouldn't get an egg, so therefore.
I mean, that's implicit in this, right?
Okay,
so that, I mean, you look at, if you've got a protein, it has some stability, it has some fold, and you want it to adopt that fold. You want it to have some minimum stability in order to carry out its function. If that changes, if it's deleterious, it's some critical interaction or something, and then it can't do its function, which might be, what you were talking about there, then yes, that means that that individual is extremely unlikely to reproduce, and they won't leave many offspring. And so I think it's effectively accounted for in the model.
So I've got a question, which is, I guess maybe it's more about the community of people working in genetics, which is, I mean, I can imagine that these are kind of the live debates between these sort of pictures of whether or not you have this adaptation and stuff. So, I mean, could you give an indication of, let's say, what the status of this debate is and how one can settle it? I mean, how does one move towards settling these kinds of debates?
I mean, as you see, lots of these things, I think it's a continuing thing, and it hasn't really penetrated very much. So I think people in the evolutionary biology are in their own camps, as it were. And so, I mean, and I think people like me who come from this physics perspective have some partial success in trying to, communicate these ideas. But I mean, this argument, and if you saw the publications, been going for 20 years. This argument of marginal stability has been around for a long time. It hasn't been expressed in this way in terms of statistical physics, but because it's very attractive. If it had, I think it depends on what. But if you're asking in terms of practicing biologists, where they just want to make some statement about evolutionary consequences, then they can very easily say, without there's selection going on, this must be selection. But I think a more practical way, I think nowadays people are asking more and more the question of are changes neutral or are they adaptive? So there is that knowledge, and that people are asking this question in the field. So have this change I see, with viruses, with different organisms, we see this change. They ask the question, is it adaptive? And they use these methods to determine whether something's on selection versus being neutral. And the standard thing is a thing called DNDS. And if you have two different lineages, you can ask what's the ratio synonymous versus non-synonymous mutations. And then what I've been trying to push with this allele frequency thing is you can also ask the same question within populations. But this broader thing that, because of this more generic genotype-phenotype map, I don't think it's, yeah, it hasn't penetrated very much as far as I'm aware. Yeah.
I mean, and then following up on that, which is because typically, many people are working on, so maybe even if you're doing modeling theory, what you're thinking about modeling some, particular process or development or something like that. And we don't usually think about evolution. So it's just always, evolution has prepared this thing in this state, and then we study this thing. So do you have anything like, I mean, how can we input evolution into the way we work? Because now we have to ignore evolution. That's just way we don't talk. So is there any way in which it affects the kind of things we study?
Yeah.
On the scale that we study, say.
Yeah, I mean, I think it's only if you're trying to make evolutionary statements, about what you're modeling. I think you need to be aware of what's the underlying, what's my null distribution. Essentially, what I'm talking about here is what's my null distribution. And if you're just trying to understand how you're modeling some process in the cell, and that's the phenotype, then you don't need to worry about evolution, because you just need to know what's the mechanics that gives me that thing. If you then want to ask how would that evolve over time and change, or if I see changes between two different species or lineages in that, the way that, maybe acting myosin networks work, then to ask the question, is that a selective advantage? You need to ask, first consider the this null distribution. Suspect most of the time you're not, though. So
One question? If not, let's